Welcome you, sir. We are very happy to have you for today's session, and I really appreciate for the entire time you have given us, though today it's a Sunday and a holiday for all of us. So I welcome you on behalf of MBSI. Welcome you, sir.
Thank you. Thank you. Thank you.
I also would like to welcome all the members for today's session. With a short time, I would like to just introduce our resource person, Dr. Bajrang Kumbhar, sir. So Dr. Bajrang Kumbhar, sir, has completed his academics from Shivaji University, Kolhapur. At present, he is working as an assistant professor in Department of Biological Sciences, NIMS, University, Mumbai. His PhD work was on effect of hypermodified nucleosides on the anticodon loop structure of tRNA. With his academic experience, he is also an experienced person from the industry. He also has played a role as a senior research associate at IIT Bombay. He has also owned membership in few associations, and his area of specialization includes computational structural biology, protein structure prediction, drug resistance pattern, and molecular dynamic simulations. He has a credit of almost 34 plus publications in peer-reviewed Scopus Index, national and international journals. So he has also enjoyed as an eminent resource person for diversified topics in bioinformatics. Also, he has presented papers in national and international conference. With all this information and a few still more to be told, I would request Dr. Bajrang, sir, to take over the session. Over to you, sir.
Yeah, thank you. Thank you, Megha. Thank you for kind introduction and very few words about me, but it's very impressive word about me. And I hope the present session will help all of you, or PhD students, faculties, or my colleagues to help in their research. So without wasting much time, I would like to start my session. And the session is highly mostly related to the computational biology, how we can use the biological data and how we can think about the drug discovery processes, what are the component of drug discovery processes, how the traditional drug discovery process and current drug discovery process helps in the, like we can say that the paradigm shift in the drug discovery process. So let me start my presentation. Just wait a minute. Yeah.
So sorry for the interruption. This is just to bring to notice of the participants that any questions regarding the session, you can either text in the chat box or you can unmute yourself at the end of the session and raise your query. Thank you.
Yeah. I'm sharing my slide. My slides are visible to all of you?
Yes, sir.
Yeah. So the topic for the today's presentation is on how to use a computational biology or biological data available on the internet resources for the drug discovery process. So why it is important? So this technique or the drug discovery process have a lot of things, which includes the having the target identification, screening the drug libraries, and finding the potential lead molecules. So how we can think about this, how we can use this data, these resources for drug discovery process. So in a broadly, you can see in the left side, there is one very important, beautiful image that which is a protein and there are drug libraries are there. And if you filter all these two components, protein and drug library, we can think about the ADMET property predictions, docking, machine learning, pen scores, and then we can find out the hit compounds. And then we can certainly go to the experimental validation without using a lot of chemical entities. We can think about the very few components of very few hit compounds for experimental validations. So it is, if you look at this image, you can say that it will reduce the time. So how the traditional drug discovery and currently available drug discovery process help in the finding the potential lead compounds. So let me tell you about the content of the presentation. So what you can learn from this today's 1 hour session. So you can get to know about the biological data, what kind of biological data is available, what are the databases are available, where the biological data stored, that information you will get, how the biological data is coming, the protein is there, protein sequences, then structural data, Then you can think about the data for the genomics data, like RNA-seq data, like DNA nucleotide data, all the data, how the data is coming and how the data is stored on the databases, which are the publicly available databases are there, and how you can fetch the data. Then I will give very broad introduction about the genome sequencing, why genome sequencing is a very important in drug discovery process. And then we'll discuss about the details about the drug discovery process, introduction, in which I can talk about the traditional drug discovery process and currently available drug discovery process. And in that you can learn about the protein structure predictions, structure-based drug discovery, ligand-based drug discoveries, machine learning, computational chemistry, and then validation by using the various approach called as the molecular dynamic simulation and in vitro study. I hope you get this content. So in addition to this, I am also going to tell you that what in my most of all presentations, all the tools, those are freely available on the internet are used. So this is very important point. The free tools you can use for the structure-based drug discovery or ligand-based discovery, means computational drug discoveries. No need to pay a single rupees for getting this use of this. Only you need a computational resources. The rest of the tools are freely available. So let me start with my presentation with a very basic of the biology, that is a cell. As of now, I can say that I can expect that you all know that the basics or basic unit of life is a cell, and cell play very important role, which consists of genetic material, amino acid, proteins are there, carbohydrate, lipids are there. So lipid bilayer, which consists of some proteins which are playing important role to send a signal inside the cell, and the cell respond to that signal and give the proper response for cell replication, intracellular transport, cell migration, cell movement. Lot of things are available. So you can say on the right side there will be a cell duplication events are there, and it plays very important role. That is cytoskeleton filamentous proteins are there. Then duplicated chromatin materials are there. That is equally shared into the two cells. So the cell consists of a nucleosides, amino acids, lipids, and carbohydrates. These are the four building blocks of the cells, and these building blocks of the cells are developed by using the central dogma. Like if you say the central dogma of life, in that you can think about the DNA makes RNA, RNA makes protein, and then protein play very important role in most of cellular process, like intracellular transport is there, cell signaling is there, enzymatic reactions are there. So if you think DNA or RNA, a nucleotide information play very important role to code the protein molecules. So how this information you can get it. So if you compare the normal cell and healthy cells, there are various changes are happening. So if you see the normal cell, so there are DNA materials are there, protein, nucleotide sequences are available, protein, lipids are there. But if you also compare the cancer cell or other disease cells, the similar kind of genetic material is available. So how you can get that information? So I will discuss first about the how to get DNA or nucleotide sequence information, protein information, and then we think about that how this information is further used for the discovery process. So how to get nucleotide sequence informations. So any infected persons or maybe you can consider the COVID-19 case today, you can consider here. So if any infected person is there, you can take a sample, isolate the DNA, then you can go for the sequencing. DNA isolation and RNA isolation are standard process. I think everybody knows about the DNA isolation, RNA isolation, basic steps involved in that. So once you have a DNA and RNA, you can send the sample for the sequencing machines. So now Illumina, PacBio, Oxford Nanoparts are available to sequence the content in that particular sample, whether it's a normal sample or disease sample. So what you are going to get? You are going to get the sorry, I just keep the pointer. So you are going to get you can get the sequences, and once you get the sequences, you can compare the sequences with the normal or healthy cells or with respect to the disease cells. So so all these information you can get it by using the sequencing machines. So we can say that WGS, whole genome sequencing, or next generation sequencing platforms are available that will help you to get what are the genomic contents present in that particular gene. Okay, so if you think about the how to get protein sequences, another infected person or any samples, tissue samples or plant samples, any bacterial samples, you can isolate the protein, separate the protein. Once you isolate the protein, you can go for the peptide digestions, and then you can go for the chromatography, LCMS, to generate the mass. And from mass peak database search, mascot is available, and from that you can identify the protein peptide. So what kind of information you will get from these two? First is the DNA sequence and another is from the protein sequence. So you will get the sequence information. So this is the first biological data you can get after the sequencing. Most of the people in experimental biology, they get the DNA sequence or RNA sequence. Where you can use these sequences, you can use this sequence to do a phylogeny, to find out what are the genomic changes that happen, or what are the amino acid composition changes are happening with respect to the protein or with respect to nucleotide. So this is the first biological data. Where this data goes, that is another question. Where this data goes? This data goes into the databases. Is it right? This data goes into databases. So once you isolate DNA or any nucleotide information or amino acid sequence information, you can put this information into the certain specific databases. So this is the biological data. Okay, how we can use this? So if you see the biological sequence statistics, okay, if you see the sequence statistics, in NCBI, that is the National Center for Biology, Biotechnology Information. So you can say in these databases, that much of sequences are available till today. So you can see in 1990, very few 10,000 sequences are available. But with respect to the time, you can say in 2021, more than millions crore sequences are available in the databases. Similarly, similar case in case of protein also. If you say in a 1996, very few proteins are available. But now if you say in 2021, 2023, there are 250 million sequences are available for the proteins. Means scientists are running behind the isolation of protein, identification of protein, and then finding what are the component amino acid composition changes in the protein, as well as if you consider about the nucleotide, then what are the nucleotide gene sequences are changing. So this biological statistical sequence data information tells us that we have a lot of information about the biological sequence informations. But where we can keep this? So with, even though you can say with respect to the year, there is an exponential growth of the biological data. But where that data is stored? So this data is stored into another biological databases called the protein databases or nucleotide databases. Now, in addition to this, there is one more data is available, and that data is called as a biophysical data. So we can say biophysical methods are there, like X-ray, NMR, or cryo-EM are there. These techniques are important to find out the protein conformation, protein structure, three-dimensional structure of protein. So you can say protein DNA RNA isolation, protein DNA RNA purification, crystallization, and after the crystallization, if you use these methods, you can get a three-dimensional structure of protein. So where this data is stored? So this data, right now there are three data. One is the nucleotide data, one is the protein data, amino acid data, and third one data is called as the biophysical data, called as the crystallographic data. So these three data divided into the three basic platforms. So one is you can say nucleotide databases, in which gene bank, DDBJ, and EMBL. These are the sole platforms where people are storing their informations. While another database called the protein databases, primary, secondary, composite, in which primary database called as the Swiss prot database available, PIR database is available, where scientists are store their data. And then in addition to this, there is a third database called as structural databases called as RCSB or PDB databases, where scientists deposited their X-ray or NMR or cryo-EM data into the databases for publicly available. So all the data are publicly available on the internet resources. I hope you all are agree with this statement. So in addition to this, in addition to 1, 2, and 3 database, there is one more database is called as a human genome database. So in case of human genome databases, called as the reference genome databases. As of now, when the human genome project is announced and it is finished, even though today, like last 1 year back, the entire genomic data is available, and all the data is stored into the UCSC genome browser or Ensemble. These are the reference databases where human genome sequence data or the genome sequence of any bacteria, fungi, viruses, all these data, reference genome data is available. So there are nucleotide databases, protein databases, genome reference databases, and structural databases available. So we have now all biological databases, and in that data is available. Now we can use this data. So how you can use this biological data and how you can take its application in a two-way. The first is a genome analysis, because we have to find out what are the genomic changes happening to the cancer cell or bacteria, drug-resistant bacteria or virulent bacteria, viruses or fungi. So you have to think about that. You can use this application into the two part. First is the genome analysis and second one is the drug discovery. So I can say we can broadly think about these applications. So hope you have a get the earlier part, that is how the data is coming in the different experimental approach, and that data stored into the databases. Now, if we can move ahead with respect to the use of these databases and find out the various applications. So first we can think about the genome analysis, and then we can think about the drug discovery process. So why genome analysis? I keep the first point because once you analyze the genome, you can get to know that what are the genomic components are vary. And once we know that what are the genomic components are vary, we can think about that. So I think there is one question: do we have any advancement in protein sequencing like NGS? So I will come to this point, Prakash. Just a minute. So in the biological, like we have I'm keeping this first point and genome analysis. So once you analyze the genome, what you are going to get, we are going to get that what are the gene changes, what are the protein changes, what are the protein composition changes, and we also get to know that these changes are important for various purposes like drug discovery purposes, or you can think about that what are the these genome changes are for finding the target identifications. So I have at this point, we will discuss more independently in the genome analysis, and then we can use this data for drug discovery process. So once you know that these are the gene changes, we can target that genes and we can find out the potential lead compounds, or we can design a monoclonal antibodies also, or small drug molecules. So let's start with the genome analysis, first part. Yeah. So in genome analysis, how people are actually identify the genome, that's very interesting. So it is called as we can say that WGS will be there. In a WGS, how you can take these applications. So in a WGS, we can think about that you can isolate the DNA. DNA extraction is the first step where you can get the entire genomic DNA from the sample. Once you isolate the DNA, you can move to the library preparations in which you have to adapter sequences will be there. Adapter is the sequences. So what is adapter sequences? Adapter sequences are important that can go and ligate to the DNA fragments, and it is further used for the sequencing purpose. So the next step in DNA genome analysis is called the library preparation, and after the library preparation, you can go to the sequencing machine. So isolate the DNA, prepare the library, and then you can jump to the sequencing. So I just already mentioned about the PacBio is there. Then Oxford Nanopore will be there, Illumina will be there. So these are the machines are available. That is the first, second, third generation machines are available. And these sequencing machine give the what exactly genomic composition are changes. So this information we can further move to the analysis part. So where bioinformatics people actually, computational biology people actually working here. So what is the data is coming into FASTA QC format, FASTA Q format, and then you can compare it with the reference genome. So BAM file will be generated when it is a reference with the FASTA QC file, and then you can find out the VCF, call it you can find out the variants will in between that. So this is the basic steps involved in genome analysis: DNA extraction, library preparation, sequencing, and then bioinformatic analysis to find out what are the genomic variations are there. So where you can just, this is actually like I will give the just more idea about that how you can take the applications. Suppose if you have a blood sample, so or tumor samples will be there. So you can take this tumor sample or any blood samples. So you can, as already mentioned about the reference genome. So in reference genomes, there is a reference genome database. Human Genome Projects find out the entire genome of the human. So all these genomes are available. So you have a reference genome and you have an individual genome sample, like either from blood sample or tissue sample, solid tumor sample. So you can find out what are the genomic components are there. You can prepare a library and you can go for the sequencing. So break genome into the small fragment, generate millions of the sequence reads, align sequence read with the reference genome. So this is the reference genome. You can take a reference genome and align your whatever the genome available in the tumor sample. So what you are going to get? You are going to get the mutation, what are the drug resistance pattern because of the change in the particular genomic component, and what are the overall genomic changes are there. So you can, this is the one of the application of the application of the genome analysis. So this is the first application, even though you can think about the bacteria. Okay, if you think about the bacteria, in case of bacteria, so where you can take applications, so you can take a bacteria, what is the sequence of that bacteria, so you can isolate the DNA RNA, prepare the DNA RNA sample, what are the genomic samples, then you can go for the sequencing and you can analyze the sample. This methodology will be the same, only you have to think about that, what are the basic which cell you are actually taking. So if you do the sequencing, your Illumina or PacBio, you can get the sequences. Where you can take the application of this microbial analysis called as metagenomics, you can say non-pathogenic bacteria are there, and then there's a pathogenic bacteria are there. So one is a virulent, one is a very shows more virulent factors will be there. So pathogenic and non-pathogenic bacteria you can easily identify, and from that, if you have a genomic component like a DNA, like a nuclear sequences, you can identify the pathogen, then you can identify the antibiotic sensitivity or resistant profile, emergence of new clone, detection of the outbreak like COVID-19 or any other outbreaks will be there, virulence factors, new genomic component present in the pathogenic, molecular datings, and global temporo-geographical distributions are there. So you can take the application of the NGS, genome sequencing, in all these areas. So this is another applications where the genome analysis play important role. And the third application, you can say application in the viral genome analysis. So in this case, you can say there is we know that in the COVID-19 start with the SARS-CoV-2. But if you, with respect to time, there are different variants are there, alpha variant, beta variant, gamma variant, and delta variant. So identification of this virus and further helpful to decide which strategy you can define. But how you can say? So you can say alpha, beta, gamma, delta variants are there. So you can identify the new strain, how this new strain is a virulent. What are the drug sensitivities? How the new variant shows drug resistant properties? What are the resistance profile, detection of the outbreak, and the new genomic components in the. So genome analysis play important role in identifying the pathogenic bacteria, viruses, fungi, or disease samples. So the overall you are going to get that the genomic changes are there. And these genomic changes, so how you can use this? So you can identify any viruses, bacteria, fungi, and will further decide the strategy for the drug discovery process. But how? That's a very interesting. I will come to the next slide. So recently we published this paper in Nature Communication, which is having impact factor 70.69. So in which we have identified the bacteria which shows colistin resistance. Colistin is the high-end antibiotics, and which shows resistance nowadays. And we have find out the interbacter species are there. So in which we find out, we collected the 300 samples from the different bacterial hospitals in Germany, and we analyzed the genomic contents, how the genomic contents are different. And now we came to know that the Enterobacter Zhang fungi is the one of the member. So this is one of the applications where you can take. The next application, we published one paper in Gene, which is with a collaboration with the ICMR Institute, NIRRCH, which is in the current level in Mumbai. So you can say we find out the novel risk factor confirming the gene that is involving glycolysis process or schizophrenia in the Indian corn species. So we find out that which gene, which are the variations are there. You can say there is a T2M variations are there. So this T2M variations play important role in schizophrenia. So we analyze the genomic content, we get the DNA, and analysis of genome consists of there is a mutations, and these mutations we further include into the protein and find out how the particular mutation impact the overall structure and function of the protein. So these are some applications where we can do a computational biology, and we can analyze the data, and we can go to the next step. So I know that in your mind there will be some questions related to the genome analysis, how we can use that. So genome sequencing, we have to, like RNA, DNA, RNA sequences, you can do at your laboratories. But once you isolated DNAs, RNAs are there, your many proteins or DNA, RNA will be there. So you can send DNA, RNA sample, nucleic sample to the PacBio or Illumina. So they will give the sequences. Once they give the sequences, you can use the publicly available free resources for the genome analysis. So the pipeline consists of a FASTA QC, BAM file, and variant analysis. It entirely depends upon the bacterial sample or maybe DNA RNA-seq analysis. What kind of DNA analysis will you have to perform? So which are the freely available softwares? You can say Galaxy. Galaxy is a freely available. So you can use Galaxy. So all the tools are available in the Galaxy. Most of the startup companies, they use first Galaxy and then they can develop their own pipeline. So Galaxy consists of a genomic analysis. So assembly, annotation, mapping, variant calling, chip-seq, RNA-seq, multiple alignment. So all these tools are available, which is a free of cost. Only you have to create the login and you can use these resources. So I also used in my initial time period that I use these databases for creating the basic steps or basic libraries for my genomic analysis pipelines. So you can also think about this. So this is for the genome analysis. So what kind of sequence information and how you can use it for the drug discovery process. That is the connection link between the genomic data and drug discovery process. So, okay. So what we have, just a minute. Now we have a sequence information. Is it right? We have a sequence information and we think about the drug discovery process. So overall sequence information is a we know that the fundamental component in computational drug discoveries. So how it is important? You can find out the target. You can identify the target protein or target DNA also. Then you can find out, once you target protein is there, you can predict the structure of that protein. Once you predict the protein, you can do a virtual screening. What is virtual screening? I will discuss this in upcoming my slides. Then you can go for the drug target and interaction prediction by using molecular docking. And then you can find out the resistance predictions. So any mutation, how the mutation impact on the particular drug. And then you can also do a de novo drug design. So all these can be done only because of we have a sequence information. No sequence information, no drug discovery. Drug discovery may be impacted or affected. So it plays crucial role in accelerating drug discovery process by guiding the selection of drug and optimizing the potential drug candidates using the computational approach. So my next slide will discuss about the drug discovery process. Now we have a sequence information. Now we can connect this information to the drug discovery process. Now, what is a traditional drug discovery? I will just focus on this point and then we'll move to the computational drug discovery. So if you think about the traditional drug discovery, so there are three basic steps: drug discovery, clinical testing, and approval for the productions. But if you think about the drug target identification, means which protein or which DNA is there, then you can lead discovery, find out the potential lead molecule that can be bind to the protein. It took around 7 to 10 years and the cost is around 600 million to 800 million. So near about 200 enter clinical trials. So after this test, we have to go to the next step called the lead optimization and preclinical testing. So all these steps, target identification to preclinical testing, it costs around 600 million to 800 million, near about. So after that, there's a phase second, third, or maybe the clinical testing will be there. It also costs 6 to 12 years and 1.2 to 1.4 billion. So it almost because 5 entering clinical testing will be there. And after this, there will be the last step called the FDA, EMA or review profile will be there, and post approval. So it is also cost 15 million, near of 50 million, and then 1 to 2 years will be there. So traditional drug discovery is taking a longer path to find out the potential lead molecules. And what are the problems with the traditional drug discovery? You can say the expensive, time consuming, that is very important. Then complex process and expertise required. That yes, we require complex process and it has required expertise are required. Then you can think about the single new drug can cost 1.5 to 2 billion and take 12 to 15 years to develop. And results are sometimes never guaranteed. Sometimes the candidates are failed. And that's the main problem with the traditional drug discovery process. And that's why any drug coming to market, the cost of that drug might be higher. So in addition to this, there are some problems with traditional drug discovery, and this includes the reduced efficiency of the drug molecule, stagnant success rate, then patent expiration is also one of the problems, then price competition from the generics, and high regulatory hurdles will be there. So how we can overcome these problems? So in last 10-20 years, there are multiple drugs are into market. So that by using the computational approach. So there is a one alternative method called as a computational drug discovery, in which there is a I think there is some question. Yeah. No. So we can think about the computational approach, and that is we can now say that is a paradigm shift in the drug discovery. So if you see the biological data, so growth of the protein databases or structural databases, millions of sequences are available, and we came to know that from the earlier session that is a drug, any identification mutations can be easily found out by using the genome analysis. Now you can think about this. In this process, we can computationally find out the potential drug molecules. But how? What are the steps involved in computational drug discoveries? So computational drug discovery involves some of the steps. The steps, before the steps, I'm going to tell you that key what are the drugs currently available in the market which are developed by using the computational approach. So you can say relative, this is called as the thymethylase synthetase, which is for the HIV, and which is obtained by using SPDD, structure-based drug discovery approach. So all these drugs are currently available in market, called against the urinary tract infection, lymphoma, cancer, tuberculosis, HIV. So these are the success cases of drug discoveries. And so you can just go to this reference, you will get to know more about this. So before spending more time on this, we'll jump to the drug discovery process. So computational drug discovery process consists of a disease-related genomic data. So you can get to know what genes, which proteins are there. You can identify the target protein. So protein structure prediction, docking, pharmacophore, druggability can be possible here. And then lead discovery and optimization, unit selection of the drug library, drug virtual drug screening, and docking and QSR. And then third step is the preclinical trials, so in which you can computationally ADMET prediction, pass, MD simulation, binary calculation can be done. So all these three are a computational process, and you can directly jump from this step to the clinical trials. So phase 1, phase 2, phase 3 will be there. So you can save your at most almost 7 to 8 years to find out the potential lead molecules. And what are the steps? So how we can use this? That's very important questions. So traditional computational drug discovery have a two important methodology. The first one is the structure-based drug discovery, and another method is called as the ligand-based drug discovery. So what exactly present in a structure-based? You have to find out the target by using either genome sequencing, or find out, if you know the target, you can target that. Then you can find out the binding site, you can do molecular docking, and you can find out the better drug molecules. So in the method, first target identification, binding site prediction, compound library preparation, docking, and MD simulation is the one way, and then you can go for the in vitro. So both LBDD and SBDD find out the lead molecules, and then you can go for the wet and dry lab and find out whether the selected candidates have potential or not. So even though it will reduce a lot of time and money, cost, expenses, everything is reduced. So I will give the broad idea about the LBDD, and then we can jump to the SBDD. So LBDD means a ligand-based drug discovery, in which, so what exactly ligand-based? In ligand-based, we know some drug molecules are there, and these drug molecules have potential as anti-cancer compounds. In which here this approach focuses on chemical properties of the drugs, on characteristics of the small molecules that will known as a ligand, and rather than it will think about the protein structure. So the principle of the LBDD is that structurally similar ligand often exhibit identical biological activities. Means you have a Taxol molecule or combrastatin or colchicine. If you have similar kind of ligand, it may exhibit a similar kind of properties. So LBDD approach focuses on ligand, not on protein, but there are multiple targets will be there. So LBDD focuses on the ligand property, not on protein properties. So how it is work? So suppose you have a ligand molecule, and similarly you can get more similar kind of compounds in the libraries are there. So people are synthesizing the derivatives, people are synthesizing similar biosimilar compounds are there. So that will come into databases. Once you have a database, you can find out the pharmacophore, then you can find out the 2D, 3D QSR, means bond donor, bond acceptor properties are there, and then you can find out which one is having higher potential. So there are, we can say it is a ligand-based virtual screening approach, and in which you can find out the similar ligands against the specific target proteins. So ligand-based approach focuses on the ligand and similar kind of ligands in the databases. So how this can be done? So compound selection. So which compounds? But Taxol, if you similar, like the tubulin is there, or catenin will be there, or maybe HIV related compounds are there. So we know this compound is earlierly shown as the higher antiviral, antifungal, or anti-cancer properties. So you can use that compound as a target set, and then you can select the various compounds from the drug library. So what you are going to get? You are going to get the similar kind of compounds from the drug library. So which are the drug libraries are there? PubChem compound is there, zinc libraries will be there, and you can get the similar compounds. So once you have a similar compound, you can go to find out the molecular descriptor properties. What is the molecular descriptor property? Descriptor property shows that how many bond acceptor, bond donor, bond molecular weight, all the properties are there. And then you can go to the similarity search, then QSAR, and design a optimizer molecules from that, and you can experimental validations will be there. So this is a LBDD approach. What are the free softwares you can use it here? That is the main agenda of the today's session, is that I will introduce you that what are the free softwares are available and how you can speed up the drug discovery process. So RDKit is a freely available. It is a chemoinformatics based upon the machine learning. So how the machine learning? We know that drug molecules and the drug molecule have a similar or some kind of a different properties are there. These properties can be done by using the machine learning. From machine learning, you can find out the active and inactive compound based upon the some kind of a algorithms are there. So this is the RDKit is a freely available. Then Chemtech is also. Then JOE Lib will be there. Then Data Warrior also. Then Chemin R, OpenBabel. So these are the freely available softwares are used for ligand-based drug discoveries. So this is for ligand-based. So might be if you have any question in between, we can talk. Okay. So this will be the second last point. So next approach, that is called as a ligand-based, structure-based approach. So first is a ligand-based, in which we are targeting only ligand, we are not thinking about the protein, and we are finding the similar kind of ligand in the databases, or you can design that kind of databases and find out whether it is effective or not. So currently, pharmaceutical industry is actually targeting, they are actually using the ligand-based drug discovery approach. Also, they are using next approach called as the structure-based. You know that in that protein is very important, and we know this protein is found to be mutated. So in that case, you can use the protein-based or structure-based drug discovery approach. So what are the steps involved in SBDD? Protein structure. You have to prepare a protein structure. Then you can prepare a drug library. Then you can do a virtual screening. Then you can go to machine learning, ADMAT, computational chemistry, and then you can validate by using the molecular dynamic simulation whether the drug is having affinity or not. So when you perform the MD simulation, you will get to know that whether drug is having affinity or not. And the next one, and very important one, is the in vitro studies. So let's jump to the one by one step. So how you can get the protein structure? So protein structure is available on the RCSB databases called the PDB databases. You can download it. If not available, you can use a homology modeling based approach. You can build a protein model by using homology modeling approach. So there are free softwares are available for homology modeling, in which there are three methods: template based fold recognition and template free modeling will be there, called the ab initio modeling, in which you can use a modeler or artificial intelligence tool to develop the protein model. So in AI, so recently there is the AlphaFold is there. AlphaFold is machine learning based, and they use a big data processing, so they can find out the fold for the protein, and it is found to be the 100% accurate computational prediction will be there. So green one is the experimental data predicted protein, and blue one is the computational prediction protein, and both are having the similar kind of a fold will be there. So you can predict the protein structure by using the freely available modeler software, which will be run on Windows, as well as AI-based tool, which is run on the Google Colab. So both are freely available. So once you predict the protein model, you can identify the binding sites, or you can find out the stereochemical quality by using the Ramachandran plot. Hope all of you know about the basics of the Ramachandran plot. Because if you build the protein and the Ramachandran plot shows that below 90%, 80% residues are in a not favored region, protein is not good qualities. So identification of binding site can be possible by using the UniProt database, SwissProt database, or earlier literature search. That is by using the protein binding site predictions. And then the next step, that is called the drug library preparations. So we know that there is a PubChem compound is there. PubChem compound databases are available on the NCBI. Zinc databases is there, or maybe you can think about the drug bank. There are multiple databases are available on the online. You can use that, or you can synthesize drug derivatives can be used here. So currently in a drug bank, you can say zinc database is available, which is the freely available. Then PubChem compound is available. Then drug bank, then IMM impact database will be there. So these are the currently available. In addition to the drug bank will be is a very important. PubChem and zinc banks are very important databases from where you can see the related compounds. You can download this compound, and you can, once you retrieve this compound, you can go for the computational drug screening. So first is the protein structure prediction. Second one is the drug library preparation, and third one is the virtual screening. So how you can do this virtual screening? So virtual screening can be done by using the free software Atodoc Vina, or maybe there are online servers are available. They can screen the compounds against the binding sites or active sites. So suppose you have a 1,22,051 like compounds will be there. So out of that, 630 is scaffold hunter, Swiss ADMT, molecular docking, and you will get only 50,000 compounds. Maybe you can say 25,000 or 10,000, or maybe you can think about the very few compound you can screen. So the screening, so the criteria for this here, the screening of the compound is that ADMT, like adsorption, distribution, metabolism, excretion properties are there. And then you can think about that binding affinity. So when you screen the compounds at the binding pocket, now the compound shows the interactions. So interactions with the amino acid at the binding point. So what you are going to get? You are going to get the binding affinity. So the binding affinity is the criteria here to find out which compound is having higher binding affinity. Means it can potential affinity toward the binding pocket. Not all the compounds are the potentially bind or show affinity, but very few compounds find to be a found to be the lower affinity. So Atodoc Vina has a Umber force field, and Umber force field play important role to find out the what is the binding affinity of the compound. So you can narrow down from millions of compounds to the very few compounds based upon the binding energy criteria. Okay, this is the virtual screening. And heat identification will tell you that which compound is having below 10 kilocalorie, below 9 kilocalorie compounds will be there. So this is the third step. And in the third step, once you have a very few selected compounds, you can use the machine learning. So it is a very interesting. I'm spent some 1 to 2 minutes on this, and then we can move to the next step. So what exactly machine learning? What machine learning actually doing? So machine learning is finding the active or inactive compounds, but based upon what? So the finding the active and inactive compound based upon the molecular properties. So you can say molecule 1 will be there, molecule 2 will be there. So molecule have some properties, CH3 here, and you can say CH3 on another point. So it is the ortho, meta, para. So sometime ortho, sometime para will be there. So CH3 will be shipped from here to there, but it changes is the 1D properties. So you can say here is the, if it is a compound is having active property, it will be denoted as a 1, inactive will be the 0. So data set, you create the molecular descriptions. Molecular description include the active compound, active like a bond donor, bond acceptor, and maybe you can think about the molecular weight, blood-brain variance, toxicity, all the properties will be there. And that molecular descriptor property can be generated by using the paddle descriptor properties. It is a freely available. So you can find out molecular descriptor properties, then you can find out the, use the machine learning model. So there are MLP, PLS, KNN, MLR. These are the various algorithms are available. You can use this algorithm and you can narrow down to find out which are the out of 1000 or out of some limited compounds, which are the active compounds are there. And that can be done by using the Weka or Python for ML. So machine learning can be like easily can be done. Nowadays, machine learning tools are available. Even though in a Galaxy, I just mentioned about the Galaxy for the genome analysis, but you can use Galaxy for machine learning also, and you will select the very few compound which are having the property is active based upon its what kind of chemical quantities are there. Okay, so machine learning is now speed up the drug discovery, and I can say that machine learning will shift the paradigm in the drug discovery process because it is an error-free process sometimes. So once you have a few compounds, like 100 compounds, you can predict the ADMET, adsorption, distribution, metabolism, expression, and toxicity predictions can be done. So you can use this Swiss ADME server. You can find out the properties, physicochemical properties, lipophilicity, drug likeness, medicinal chemistry, pharmacokinetics, water solubility. All these parameters you can predict it for that particular compounds, and from that you can easily find out which compound is following the proper, like GI absorption with a high, means good molecules will be there. Molecular weight, how many rotatable bonds, how many H bonds will be there, that can be easily tracked from this. Then once you find out of 100 only 10 compounds or 5 compounds are there, then you can do computational chemistry. You can find out the LUMO and HOMO, higher occupied molecular orbitals, lower unoccupied molecular orbitals, means molecules having the occupied orbitals that can be used to share the electron or accept the electron. This is a very important property that can be done by using the computational chemistry. So again, you can see Orca and Bibo view can be used here. These are the freely available. So to predict the properties of a molecules or any drug molecules. So this is the fifth step, and after the fifth step, you can validate by using the in silico. So what exactly in silico? In silico, you can use molecular dynamic simulations. So RMSD, so binding affinity, then stability of the complex can be done. So again, here Gromax is a freely available. NMD, Umber is now also free to use for the in silico approach or MD simulation. So what is the MD simulation? In which we can predict the similar environment as observed, as we can do in vitro and in vivo. Similar kind of environment you can create. So these are the water molecule. This is a protein with a drug molecule, and you can simulate the molecule for a certain period of time. You will get to know the molecule having expel from the binding pocket, or it will be remain stay and have some kind of a interaction. So that can be interaction can be studied by using the all these plots, and binding affinity can be further studied. Next one is the in vitro. How you can validate in vitro? In vitro, you can say lab testing will be there. So MTT, soft agar assay, lymphocyte assay, sphere forming assay, wound healing assay, VI matrigil assay is available using the in vitro. And then in vivo, you can do in animal model, test for efficacy, test for toxicity, test for tolerabilities, and determine the PK. So in this way, computational in silico may nowadays it is helping to speed up the in vitro and in vivo process also. So within a short period of times, you can identify the potential drug molecules. So what is MD simulation? You can say there are some of the movies in which the DNA interacts with the protein, and in which the drug is interacting with the protein molecules. So it gives the idea about that how the protein and drug interactions are feasible or not. So protein drug interactions, pro-drug resistance pattern, identification of potential drug candidate, effect of mutation on drug molecules or DNA molecules can be easily studied by using the MD simulation approach. So these are the some application of the MD simulations. So you can just say there is a one movie in which one drug is highly bind to the particular protein, while in this drug is smoothly removed from the binding sites. So this indicates that this has higher affinity, but this has a lower affinity, because MD simulation, this is the advanced MD simulation called as a steered MD simulation, in which you can pull the molecules from the binding sites. If the molecule take more forces to pull the molecule, which indicate that molecule having higher affinity. In case of another molecule easily pulled from the binding pocket, it indicates that molecule have a lower affinity for the protein molecules. So as I have mentioned that MD simulation give the binding affinity, you can say you can find out the van der Waals, static interactions, and overall binding energy. From that, you can find out the binding affinity, further help to understand which molecules. Suppose you have 5 molecules. Out of 5 molecules, the molecule number 1 shows higher affinity. You can consider that molecule for the in vitro and in vivo study. So these are the application of the MD simulations. So the case study, then you can just say we published this paper in JBSD. So in which we find out the anti-diabetic drug molecule. Out of that, which anti-diabetic molecule could be act as a potential anti-COVID molecules. So binding affinity indicate that the molecule like ELUF, like EL6LU7F molecule is having the higher affinity. Means EF, this molecule has a higher affinity toward the anti-COVID, like COVID protein called as the main proteases. Similarly, I have earlier publications in that area. So all these publications I published from IIT Bombay, while this publication is from the NMIMS University Mumbai. So this publication is on the tubulin and how tubulin shows the drug resistance pattern because of the variation in the binding affinities. So currently we are targeting the tubulin. We are working on urobilin, paclitaxel, vinblastine binding sites and finding the potential drug molecule by using the structure-based drug discovery approach. So last but not least side. So what are the application of the computational drug discovery? So in which you can screening of an extensive drug library and find out the potential drug libraries. From that, we can identify the potentially biologically active drug molecule which have a affinity towards the particular target protein. Even though you can use the LBDD approach, you can find out of 10 millions of the drug library which are similar to your drug library, which drug is have potential affinities. So another application of this is a drug repurposing. We can possibly against the various disease conditions. As I have mentioned here, we use this anti-diabetic drug, and we can think about anti-COVID. So drug repurposing is the only approach right now for the viruses. It is observed that in a COVID situation, there's not a single drug is available for the anti-COVID, but most of the drugs, other repurposed drugs are there, and that can be done possible by using the computational drug discovery. So I keep this some of the slides for you. Maybe out of here, some of the students are actually PMSc or maybe PhD students are there. If you have this sound knowledge in this area, you can think about your career in the pharmaceutical industries, cancer research institute, genomic proteins laboratory, vaccine synthesis companies, and academics also. So what are the companies are available? You can just say these Inoplexes, BSET, DBSF, Presence in Silico, Scilab Sciences, Engine. So Schrodinger, Think Molecular, these are the some of the companies from the India. They are actually hiring the students, those who have a basic knowledge in the drug discovery process. While so this is the last slide. So I am thankful I have a team member like Purva and Prutanka. These are the two PhD students working on the different approach. So Prutanka is working on the structure-based drug discovery for the tubulin, drug-resistant tubulin isotypes, while Purva is working on the CAR-T therapy for the cancer treatment. So I would like to convey one more message. Currently, I am working at NMIF universities, and now in our universities, the admission is open for some of the courses, like a BSc in Biomedical Sciences. This is 3-year programs, and the next one is MSc in Biological Sciences, is 2-year programs. In which the last semester is for the project based. You can go outside for the projects, and now the admission is open for these programs. In addition to this, there is MSc in chemistry. So here also the admission is open. For more details, you can log on to the science.nmims.edu. And if you have any query related to that, you can log on to this particular website and just go to the admission process. And thank you very much. I hope.
Thank you so much, sir, for such a nice, wonderful session.
Thank you.
Thank you, sir.
Yes.
We'll take over the Q&A session. If anyone wants to unmute yourself, you can raise your questions. So the first question. Yeah. Please.
So this is Gupte, sir. I'm pursuing my PhD from Manipal University, Jaipur. So I've just started off my PhD. And basically, sir, what we are working on is we are working on finding an inhibitor against an oncogene. So we've targeted one oncogene. Say, for example, we are targeting androgen receptor, variant 7, and we are finding a inhibitor. So what my guide, his way of working is, he's directly asked me to work on GROMAX.
Directly asked to work on
GROMAX. So we have eliminated all the steps from analysis, genome analysis, then sequencing, everything, all of that we've cancelled out, and we're directly going for GROMAX approach. So right now, what he has asked me to do is take structures from PDB and then try to equivalate the structure and all of that.
No, ma'am, I think, can I stop you?
Yes, sir.
So this is completely, I think, say GROMAX is for molecular dynamic simulations. GROMAX will not help you to find out which are the drug molecules are there. So you have to think about that GROMAX will come at the end. It is called as it is used for the validation of the what are the drug molecules are there, and if the drug molecules have some affinities, you can select some of the drug molecule and then you can go for the MD simulations. So before the simulations, you think about the protein and drug. You have to find out the potential drug molecules from that. Is it right? So maybe you can use the structure-based drug discovery approach, and then you can find potential lead molecules from the earlier drug. Like maybe you can think about the earlier drug libraries are there. You can think about the drug library, find out potential lead molecule, and then you can jump to the MD simulation.
Yes, sir, I got your point. Sir, also, you were talking about synthesizing drug derivatives. So how do you, traditionally, how do we approach to that?
Yeah. See, suppose some of the drug molecules are there. Suppose you can take the Taxol. Taxol is highly used for the cancer treatment. So you have a Taxol. If you take the Taxol for consideration, and you can find out, you can synthesize derivatives. So there are a lot of organic peoples are there. They can easily synthesize the derivatives of that. Once you have a derivative, out of that which derivative is found to be the highly effective, that can be studied by using the computational approach. You can do a molecular docking, and you can find out key, we can screen down that case, narrow down to the which drug, which derivative is found to be highly effective based upon the binding energy calculations. And then you can do a MD simulation out of the 3-4 compounds and find out which one is effective, and then you can use that compound for the experimental approach. That can be possible.
Okay, so and so lastly, is there, do you recommend any specific book or to learn or to like to go about with drug discovery? Any specific book, like we have Lehenger for molecular biology, so similar to any similar book for drug discovery?
I have a book. I will send you to the Megha. Maybe she will send you, circulate the same thing to all of you.
Sure, sir. Sure, sir.
Yeah, I have.
Thank you so much, sir. Thank you
Yes.
so much.
You are from which university?
Manipal University,
Manipal University. Okay.
Jaipur.
Okay. Yeah.
So
Thank
the
you, sir.
next question is computational biology a part of synthetic biology?
Yeah, you can say computational biology will help you in a synthetic biology. Yeah.
So the next question is again from Prakash sir. He is asking that most of the freely available softwares have limitations with reference to handling of high molecular weight biomolecules. Is this true or false?
False. You can use it.
Okay. Next is from Ayushi Malik. How can we create 3D features of binding affinity for proteins?
3D features for binding affinity of protein. Features
Yes,
means
sir.
what? So means interactions.
Yes, sir.
Interactions can be, so if you see the interactions of the 2D, like a van der Waals interaction, electrostatic interactions, or maybe you can think about the bonding and non-bonding kind of interactions, you can use the freely software. Like you can say Discovery Studio is available, then Schrodinger suite is available. It is a free suites are available, visualization suites are available. You can do it, or otherwise you can use the PDB sum. PDB sum is the online server will be there. From that you can put the data, protein ligand, and you can find out which are the interactions are available there. That can be done.
Okay, sir. The next question is again from Prakash sir. He is asking how to identify specific target against a known peptide antibiotic.
Known peptide antibiotic.
Yes, sir.
So target identification can be done by using the various approach. Either if you have a sequence related information, or target identification can be done by using the various. You either first you can think about the literature search. So what are the most possible proteins or genes involved in that? So gene ontology is the one way that you can find out which are the genes are highly expressed in that particular data set. Gene ontology is there, or literature search will be there. These are the two standard approach people are using. Or third one is the NGS. We have a protein like your sequence, genome sequence is available. From that also you can find out which are the most possible target genes are there, called as the hub genes will be there. So there is a very new approach is there called as the network pharmacology. And in network pharmacology, people are finding the possible target genes. So I think Prakash, you can just think about the network pharmacology will help you to find out the most possible hub genes.
Okay, sir. The next question is from Ayushi Malik. Can you suggest some softwares for genetic pathway or interaction analysis?
Genetic pathway. I just mentioned the gene ontology, but right now I can't give the answer proper. But I will send you the details to with the same person. Okay.
Sure, sir. Still any more
There is
questions?
one pathway. There is one suit is available.
Okay.
KEGG, you can think about that. Once you know the genes, then you can find out KEGG or gene ontology. GO will be there, or gene enrichment analysis can be done.
Okay.
Enricher is the software.
Okay. Okay.
Thank
Ayushi
you so
Malik,
much.
yeah,
Yes, ma'am.
you have any more questions?
Yes, ma'am. Thank you so much, sir, for answering my questions. And my question was regarding you showed some proteins and their ligands are binding, and some ligands have different affinity, so they were getting separate. So how we can show these features in our work? Can you suggest some softwares for that too?
Can you refine your answer question?
So you showed protein ligand binding, and then you showed that this protein had had not that much activity against this ligand. So this ligand got separated. So you showed whole activity regarding that. So how can we show these activities in our things?
Computationally?
Yes,
See,
sir.
what exactly the basic idea behind the computational drug discovery? You should know the binding pocket. Okay. Once you know the binding pocket, so binding energy is the criteria. So binding energy, if the binding energy is a very high, means negative, -40, and one compound is the binding energy -10. So which one is the more stable? -40 will be there. So binding energy is the criteria where you can keep you can think about that. Once you the binding energy is very lower, -40, -20, -3. So binding energy is the only criteria where you can select the compounds. Rest of the compound you don't care about that because they have a very less affinity toward the target protein.
Okay, sir, but how can we create these 3D features in them, which you created?
Just I will show you this. No, I will sharing my PPT and then you can show me that which one is that you are actually talking. This one?
No, sir, previous.
Just so I just started with structure based. Okay. So this is homology modeling, then protein. This is the drug library. This property?
No, sir. You showed two protein comparison.
Have you
Yeah.
noted the
Just a minute.
slide
Just a
number?
minute. Yeah. Just a minute. Just a minute. This one.
Sir, one in dark view, and you showed binding site 2 in that.
Just a minute. I know that.
Yes.
This one, protein adenine binding sites.
No, sir, after that.
After that,
Sir, similar to this green one. You showed two pictures.
Sir, I think it's
And
the
one-
movie that you had shown. There was
Yes,
a
sir.
there was a movie.
yeah. Just a minute. This one, na?
Yes,
is
sir,
a-
this one.
Yeah. So see, this is a molecular dynamic simulations. So in molecular dynamic simulation, it is advanced simulation. So what exactly we did, we keep this protein and we dock the ligand molecule there. And after that, we use a steered molecular dynamic, SMD will be there. So when you suppose any weak person is there. So if you pull the weak person, what will possibly it will easily come to you. And if any heavy person, strong person will be there. If you have to apply more force to pull that, means if you see in the first the drug molecule, you can just see here. Laser pointer, you can say this one take a lot of time to come from the binding pocket, while in this case this protein you can in this protein and drug is easily expelled from the binding pocket, which means that the binding pocket is a very less efficient to hold these drug molecules, and in this case the binding pocket is much enough to hold these drug molecules. So this can be done by using them after the molecular dynamic simulation. So you can use SMD for this structure based steered molecular dynamic simulations can be done.
Thank you so much, sir.
Yes. So if you need any help in this regard, So I am going to put my email. I will, we can talk that also on that. So we'll help you in that.
So I have one thing to ask. So there are some oncoproteins that lack this binding pocket. They do not have the binding pocket. For example, MYC. And MYC is expressed in most tumors. 70% of human cancers have MYC is overexpression. And MYC also lacks this binding pocket.
Lacks binding pocket. Na, see,
Yes.
So a thorough literature study is very important in most of the times. If you have a protein and a protein is playing important role in some of the genetic pathways, plus signaling pathways, or maybe it interact with another proteins. So how any protein inhibit the binding with another protein, or how you can stop this functionality, that can be studied by various approach. Either mutation is the one that protein induces some of the mutation, and finding the better inhibitor for that, that also play important role. So inhibitor finding can be done by using the various way. So active site predictions or binding pocket predictions can be done by using the various online softwares. So you can, online servers are there. So identification of the binding pocket can be possible by using the online servers are there. So pocket predictions are, so I think I just share binding pocket prediction tools are there. Okay. Predicting, I am going to share one paper that is on the predicting the binding site from unbounded versus bounded protein structure. So you can just read through this. You can just go through this paper. And also there are some servers are there. Bind web server is there. Also, I am going to share that with you. So you can just see this paper will help you predicting the binding site from the unbounded state. And another is the bind web server or deep site server will be there. That will help you to find out where is the binding pocket, is any binding pocket is available or not. See, not most of the time you can think about the bigger ligand molecules. Even the small ligand molecule also inhibit the protein structure. Like a thymol is there. Thymol is present in the OA that we use for the household, like ingredients are there with the kitchen ingredients. So we can use it. So in OA there is a ajwain. In ajwain there is a thymol is there. Thymol is a very small compound, but it can potentially do lot of good activities. So if you find out any small ligand, you can find out the better potential affinity, and you can do any vitro studies. Maybe you will get a good ligand molecules.
Thank you, Bajrang sir.
Yes.
Thank you so much, sir. Thank you.
Yeah. I have given my email ID. So even though I can share on my screen. So anybody is interested, you can see my slide now.
Yes, sir. It's visible.
Yeah, I can put some email ID here. So anybody is interested, you can.
Sure. So I guess the questions are done. So the last one is a very important one. So I would like to really thank Dr. Bajrang sir for such a wonderful session, which was knowledgeable in all the ways, from the basic till the end. And I would like to thank you more for creating awareness about computational biology, about drug designing by computational biology, and about creating awareness with respect to the softwares that are freely available for us, because many students and scholars who are doing the research are not still aware of what are the websites that are available or the applications that are available free of cost for doing all the research work in a higher way. So I would really like to thank Dr. Bajrang sir for giving us knowledge regarding all the aspects in computational biology and regarding the free softwares that are available, and also for spending his valuable time, though today it's Sunday. I would also like to thank Deshmukh sir.
Though today is a Sunday, still I am in office.
Yes, I could see that. That's what the spirit of working, and people like you will inspire all the research scholars, sir. So that's really a great inspiration for all of us. I would also like to thank our Dr. Deshmukh sir, who is always a support for creating such awareness about opportunities about the research in life sciences. And last but not the least, I would like to thank all the members who are really actively participating in today's session, and I hope have enjoyed the session. So thank you all of you once again. Thank you, sir.
Mega, we'll take one photographs. Keep everyone to ask.
Participants, can you please switch on your cameras?
Yeah. So how we can keep in a gallery view? Yeah, gallery view. Yeah, that's great. Thank you all of you for joining on Sunday. I hope you enjoyed the session.
Yes, sir.
Yes,
That is
sir.
for sure.
Yes, sir.
Prakash sir, how are you?
Oh, fine, sir. I am actually from CFTRI Mysore.
Yeah.
I already sent you email. I will contact you later.
Yeah. Thanks.
Yes.
It was nice to have you, Prakash sir, for today's session.
Thank you. You are from which department in Mysore University, by the way?
Microbiology, sir.
Okay.
I'm working under Dr. Mahadev Murthy.
Mahadev Murthy is in Yuraja College.
Yes.
Yes. Oh, wonderful.
So what actually I am planning to keep such kind of a pre-session to all PhD students. So today I give a broad view about what exactly computational drug discovery means. But if we keep the free session for small part, like only protein design, then small molecule docking, analysis, then it will be the great help for, maybe it will be important for all you new generations. So
Yes,
you can
sir.
jump to the next level also.
Truly. And sir, actually, what I have found is still, even after the PUC, students and parents are still running behind engineering and MBBS and doctors, but they don't know the real value of being in life sciences with respect to all this upcoming trend, which is very important. So it will be better in creating awareness, not only for the research scholars, but also to the next generation, where all others will be the parents and they can pass on the same knowledge about this to the society. So it is not only going to help the scholars, but also create awareness about opportunities in life sciences.
And one more thing, that I am hardcore chemist. I did my BSc in chemistry.
Yes, sir.
And then MSc in biochemistry.
True.
And from my PhD, when I joined as a PhD programs, at that time, I don't have my mobile phone also. I got my first mobile phone in 2009. And I registered in PhD in 2007.
True.
So I first started using the computer, and then I started using the mobile. So technology is very important, but how you can use technology is one of the important things. And nowadays, a lot of resources are available online. Only you have to stay in front of the machine and spend valuable time. You will get more on that.
Truly said, sir. So as you have said, we will really try to keep sessions whenever possible from your side, according to your convenience, with respect to computational biology and bioinformatics, so that it can help all the research scholars in their upcoming research fields. So we'll make sure we'll plan out something for this and work on the same.
Yeah, sure.
Sure, sir. Thank you.
Yes.
Thank you, sir. And thank you all the participants for joining and making this event really worthful. So can we end the session, sir?
Yeah, we can.
Sure.
So I have sent my email.
Yes,
okay,
sir.
so I think there is some mistake in my email. You can just capital is not a capital B, it's a Bajrang Kumbar at nmims.edu. It's not capital B. I think people will send wrong email to them. Yeah, it is the right email ID. Okay.
Okay,
please.
sir. Fine.
Yeah.
Sure. Thank you, sir. Thank you for your valuable time. Thank you to all the participants.
Yes.
Shall we end the session, sir?
Yes.
Yes. Thank you.
Okay. Bye.